circulating tumor cell chip consists of a series of micron-scale channels that are useful for separating white from red blood cells. This process expresses the ability to distinguish circulating tumor cells in blood from a cancer cell. The effectiveness of this segregation of blood cell from tumor cells can give rise to an early cancer diagnosis.
The first part of your abstract said you're separating white from red blood cells, erythrocytes from leukocytes, basically. Given that every part of the body, including tumor cells, has white and red blood cells associated with them, what I'm not understanding from the abstract is how does it specifically delineate tumor cells?
By a certain shape and size of it. It's run through a series of channels, and so like they're kind of knocked off to the side. There's like little pillars, and they're sorted according to like the size of the cell. The red blood cells waste, and then there's a problem between the white blood cell and the tumor cells because they're very similar in size.
Your idea is very, very good. That would be an excellent invention, in that you could just take a sample and put it through your particular apparatus that you just described and understand, one, if it was malignant, and two, maybe even the type of cancer based upon the research that exists for how cancer manifests itself in the size and shape of different white blood cells.
A circulating tumor cell chip comprising sorted cell segregation of blood and tumor cells, separation of blood cells from tumor cells by movement through sorting channels, and a cancer diagnosis by using a blood and cancer cell sorting chip.
That's a very, very good claim.
Isolated tumor cells are used to monitor cancer, but are very difficult to separate from blood samples due to their size and rarity. This invention is a medical device which comprises the sorting of cells from whole blood samples by passing through a series of chambers and isolating rare CTCs into an individual compartment. A medical device comprising 4 chambers to hold sorted cells and microposts spaced far enough apart to let normal cells pass through while directing rare circulating tumor cells to their own chamber. A blood sample is dropped into an opening and micropost direct cells into chambers according to size.
In your specification, you would need to explain that the tumor cells that you're trying to isolate are of a larger or smaller size than other cells. So, and you would want to put that scientific support into your specification to support your invention.
The present invention is directed to a machine which will perform a procedure for detecting cancer cells, for detecting cancer from the viscosity of pleural fluid. The machine is called the viscosity detectotron. The machine is able to take pleural fluid extracted from a patient and determine the fluid's viscosity. The machine will do this by dropping a tiny glass bead into the fluid and using the splash created by this bead to determine pleural fluid viscosity. The splash is recorded by a camera within the machine and then analyzed by an internal computer. The machine will be able to do a procedure normally performed by lab technicians, but faster and more efficiently.
Good, very good. So the hypothesis then is that the more viscous the fluid, the more likely there is cancer.
Cancer cells.
Correct. Okay. Great.
A machine which will perform a procedure to determine the viscosity of pleural fluid, to determine if there are cancer cells within that fluid. Using a new method, a glass bead will be raised to a certain height and then dropped into a vial of pleural fluid. B, the splash produced from the bead is then recorded by a camera within the machine. C, video of the splash from the camera is then analyzed by an internal computer which can then determine viscosity of that fluid. D, higher viscosities of pleural fluid indicate presence of cancer cells.
If you were actually to write that as in a patent application, you would want to, in your claim, give the parameters for the viscous measurement and talk about the scientific research. You would also want to talk about the precise point where you're having extraction of the pleural fluid.
This software is meant to make the process of pattern recognition in the forensic field much easier. The software uses 3 unique image analysis tools to further describe the pattern of an unknown substance. The resulting data will then be compared to results of known substances from the accompanying database to then determine what the unknown substance is. The software and accompanying database will include many substances, including various blood types, to further assist forensic scientists in their field.
Assist forensic scientists in doing what?
It could be used if there's like a spill of some substance they don't know what it is, then they can use the pattern of how it dried and compare it to substances in the database.
How is this more advantageous than like gas chromatography or HPLC or some other chemical tests?
We're dropping them and we're seeing how they dry, taking pictures of them, and then we're feeding that into this software to get the database. And then we're kind of backtracking using like an unknown solution and seeing if the software can detect what that solution is.
An on-site analysis,
right,
without having to send it back to the forensic lab for gas chromatography or
right,
HPLC, right, where you could take the camera, take a picture, feed it into the software, and then have instant analysis.
Without having to burn it to get the gas chromatography.
Yeah, still preserved. A pattern recognition software comprising of a way to input an image of the unknown substance, a means by which the image will be analyzed, an output mechanism to give textual results, and an evaluation process by which the results can be compared to known substances in the given database.
Describe the evaluation process in a little bit more detail to really prove why it's novel and non-obvious.